Good morning. My name is Roger Demikowski. I'm a member of the scientific committee for the ICS, and it's my great pleasure today to introduce to you our state-of-the-art lecturer who is extremely well-versed in the topic to be discussed. Bertil Blok is our state-of-the-art lecturer this morning. Bertil did his medical training at Erasmus and then subsequently acquired a PhD at Groningen in neuroscience, and for 9 years was a researcher in neuroscience, and then made one of those very difficult life decisions to go back and become trained in urology. And I can tell you from personal experience, those are extremely stressful but courageous decisions, so I commend him for that. So he went back home to Erasmus to complete his urology training, and now he did his urology training at Academic Medical Center in Amsterdam, and then went back home to Erasmus, where he is now director of ambulatory services at Erasmus. He has established a true reputation as one of the key thought leaders in neuroscience around the world, again, because of his back history as a neuroscientist. He also is chair of the EAU Committee on Neurourology from the guideline standpoint, so is well-versed in this topic. So he will be discussing today CNS mechanisms in voiding dysfunction. I'd like you to welcome Bertil Blok. Bertil?
Thank you very much, my colleague Domachowski, for the introduction. I would like to thank all of you coming at this early hour. It's difficult, but I appreciate that a lot. And I also want to thank the organizing committee, especially Professor Yoshimura, for inviting me to say a little bit, talk a little bit about the CNS mechanisms in voiding dysfunction. My disclosures. Medtronic helps me with a prediction model on pelvic floor dysfunction and axonics. If we talk about the neural control of the urinary bladder, there is much increased knowledge by better techniques in the last 20 years. What I will show you is that it was the technique which led the progress in knowledge. Mechanism of action of central bladder control is much better explained in my view. And people say many times the brain is very difficult to understand. I do not agree. If you take a modulatory view on the brain and separate it in several separated pieces, it's much better to explain what's happening. And unfortunately, the science was not aimed at improving treatment, and that's why it didn't have any clinical or hardly any clinical relevance. I will talk about how to achieve, in my opinion, more clinical relevance in the next 5 years. Around 80, we had some knowledge about the physiology of the lower urinary tract, but we didn't have any knowledge about the specific anatomy, because degenerating tract tracing was with lesioning method and the silver method from Nauta, Gigax, and Finkheimer, and only terminals were visible from large myelinated fibers involved in somatic control, like the corticospinal tracts or the corticobulbar tracts and the ascending spinothalamic tract. Small fibers from the limbic system or involved in autonomic control were not visible. This is an example of those lesion experiments from Hans Kuipers, the former head of anatomy in Erasmus MC. And this was a paper in Science, and he showed that only higher primates have direct projections to the motor neurons of the fingers. So these projections were responsible for the individual finger movements, and it explains why when someone has a stroke in the anterior, the artery cerebri media, that he doesn't have any individual finger movements anymore. So the neural bladder control around 1980 was like this a little bit. At the caudal part here of the the caudal spinal cord, you had a sacral micturition reflex center, physiological determined, but no one knew how exactly this, the anatomy was of this sacral micturition reflex center. I don't use the term anymore, sacral micturition reflex center, because in itself it's not a full micturition center. And also from the higher regions, we knew there was some inhibitory action on the pontine micturition center, and from the midbrain, inhibitory influence, and from the cerebellum, but the exact nature of these connections, no one knew that. Then something happened after 1980. There was a new technique. The introduction of horseradish peroxidase, radioactive leucine, and other compounds with which you could also track small fibers and their terminals. And now the connections of neurons involved in autonomic regulation were visible from the hypothalamus, limbic system, periaqueductal gray, and other areas. And there was also a new thing which was coming up, the combination of physiology and anatomy. For example, Chet Groth and his group already in 1978 showed for the first time that the connections from the bladder, the sensory connections, terminate in lamina 1 and the intermediate zone of the spinal cord, the caudal spinal cord. Also was shown the exact location of the parasympathetic motor neurons and the motor neurons of the pelvic floor in Onuf's nucleus. Gert Holstegens, together with Derek Griffith, they showed a combination of physiology and anatomy. When you stimulate in the pontine micturition center, it's in the arrow, you get a full-blown micturition as shown here. Every time you stimulate, first the pelvic floor relaxes, followed by a contraction of the bladder. When you stimulate in another area, more laterally localized, every time you stimulate, you get a forceful contraction of the pelvic floor, no action on the bladder. The micturition response to the pontine micturition center could be explained by the projections to the sacral cord and the projections from the more lateral area, now known as the pontine storage center, could be understood by the connections to the nucleus of Onuf. Later on in Groningen, we explained how it exactly works at the sacral cord. We stimulated the sacral cord, the medial part, and we get a relaxation of the urethral sphincter. And we showed the connections from the pontine micturition center to the intermedial medial cell column goes via GABA and glycine, which these neurons project to the Onuf. So the whole circuit at that time was pontine micturition center is excited, an excitatory signal goes to the parasympathetic preganglionic motor neurons, bladder contraction, but at the same time collaterals go to inhibitory neurons, which in turn relax the urethral sphincter, and full-blown micturition can happen. Afferents, Chet Groth together with Noto described that physiology, when you stimulate the pelvic nerve, you get short latency evoked potentials in the periaqueductal gray. We showed that from the sacral cord that there's a specific projection to the periaqueductal gray and not to the pontine micturition center in the cat. In that sense, the loop was known. Basic micturition reflex goes from the spinal cord, afferents go to the periaqueductal gray. When micturition can take place, an excitatory signal goes to the pontine micturition center, and full-blown micturition can take place. Then something happened in around 2000. Again, a new technique, and that helped enormously in explaining how it works in humans. Dynamic imaging, first the PET scanning, later the MRI 1.5 Tesla, and again later pre-Tesla, which is now the state-of-the-art in most hospitals of the scanners. And again, it was aimed at the description of mechanism of actions. For example, we published that when you put someone in the PET scan, you get the excitation in the pontine micturition center of the human. At the pons, you get excitation of the periaqueductal gray, and in the hypothalamus, and in the cortex, in the anterior cingulate gyrus, and the dorsolateral prefrontal cortex, both parts of the so-called emotional motor cortex. Here it's shown on the left, pontine micturition center during micturition, and on the right, the lateral region, the pontine storage center, or the L region, in people who could not void but were trying to help the investigator, but the system says, the situation is not safe, squeeze your pelvic floor. This was also done by Atwell in another fashion. They filled, together with Claire Fowler, the bladder, and they showed also that the periaqueductal gray during bladder filling is activated together with the cingulate gyrus and the dorsolateral prefrontal cortex. Later on, other PET studies and functional MRI studies more elaborated these findings, much more refined than the initial publications, and big area of the anterior cingulate gyrus seems to be involved in bladder modulation, and also the pons together with the periaqueductal gray, and some other motor areas which I don't think are really involved specifically in the micturition reflex. The cerebellum, for example, the supplementary motor area, and the thalamus. Another area was found mainly in women, but also in men in the insula. Recently, Ria and colleagues published a meta-analysis, it was this year, of 180 subjects. In all those studies together, 14 studies, and they showed what the common activation is. And they described the thalamus is activated, the right insula, the right periaqueductal gray, the rostral pons, and the cerebellum. I don't think this is an important paper, but this is exactly what the problem is with the dynamic imaging now. Because what you do is you don't look at an individual, but you lump up all the activation in one brain and you say, oh, this is the common activation. Because if you look at the periaqueductal gray, that's not really in the periaqueductal gray, it's more the tegmental area, the mesencephalic tegmental area. And if you look at the rostral pons, it's not really the location of the pontine micturition center, but it's more the pontine tegmentum, or even a part of the Raphan nuclei. And the same for the thalamus. I really can't explain that, because it's a medial thalamus, mainly involved in cognitive actions. Also, studies have been done on patients, usually in very small study groups, several studies, no control groups. They use always uncorrected activations, that means low activation, and you can freely manipulate with the statistical facts, and in my view, little clinical impact. But the authors were, of course, the first one who tried to visualize clinical problems. For example, Derek Griffith recently published the effects on physiotherapy, and they showed that when you have responders on physiotherapy, first you have an immense cortical activation. After training, you have much less cortical activation. And in the non-responders, you don't have such a huge decrease in activation. So physiotherapy has an important effect, I think. We showed with sacral neuromodulation, with the PET scan, when it's first switched on, that cortical areas are active, involved in micturition control. What are my conclusions from the dynamic imaging in the patients? Activation is blurred out when you summate data from subjects into one normalized brain. Neuromodulation works similar in all modalities, in my view. If you talk about pelvic physiotherapy, tibial nerve stimulation, sacral neuromodulation, or deep brain stimulation. In my view, the mode of action of neuromodulation in those modalities is first modulation of cortical areas. New cues result in alertness. After learning, you have activation of more subcortical areas. Old cues, learned behavior. And if you look at neuromodulation, you must realize this is a picture from Tyler and Polesic, and it shows the risk contra the benefit expected. When you have a low risk, for example, physiotherapy, or here, PTNS, you have a very little benefit relatively to the other modalities. When you do deep brain stimulation, you have an extremely well benefit, but a very high risk. And people who suggest that you should implant deep brain stimulation for incontinence, I think it's ludicrous, because I would ask always myself, would I like to have that with the risk of an abscess or a stroke to get it implanted in my brain? The answer is no for myself. So when the answer is no for myself, I don't do it in my patient. Sacral neuromodulation, I think, has a very well balance between risk and the benefit expected. Here is shown the spinal root activation is exactly in the middle. But again, physiotherapy or DBS, they all work in the same manner. What's the problem nowadays with the functional MRI in 2015? Too low resolution to make a clinical impact, only results from group analysis, no validated biomarker for bladder dysfunction, not applicable to individual patients. What to do? I think it's important to realize when you are a scientist that you must go from passive observation, like the studies just described, to active intervention. And this is a quote not by myself, but by Gero Wiesenbrock. That's an inventor of the optogenetics, and we use optogenetics also at our lab, and I think it has revolutionized neuroscience. When you put on a light on a neuron, very easily you can excite that neuron and study the behavior of the subject you want to study, whatever the task. I quoted them in Wire this year. If you look at the progress of the functional MRI, this is from Menon in 2015. In 1980, you had 1.5 Tesla, 1.5 Tesla, and you see an exponential growth to higher resolution. But it's the typical S-curve. First, it grows exponentially, and now I think we are in the end of the evolution. We have at our lab Tesla 7 functional MRI, and I want to tell you a little bit about that, our experience with that. It gives you significant activation in individual subjects, higher spatial resolution, more exact location of the activation, and probably more clinical relevance in the end for diagnostics, evaluation, and prediction of possible therapeutic effect. Most of the studies are done in this fashion. You put a coil on somewhere of the brain. It gives you an excellent, this is an example of the occipital lobe, visual cue, beautiful resolution, but you lose 95% of the brain. So all the studies so far, or almost all of the studies so far, which have done in the world with 7 Tesla MRI, are done in this fashion. We choose to use the whole brain method. It means less resolution, but a wider view. I always like the idea of using a shotgun in science and then go down, tune in on the detail, not the other way around. I think that's not a good method for myself. So use the shotgun, and for example, this is an example of pelvic floor excitation. You see the motor cortex activated here as cerebellar areas, and very fine you can see that the whole brain is covered. We did for our initial experiments tactile stimulation, genital, repetitive pelvic straining, and tactile stimulation of the medial part of the foot. I thought of PTNS, so why not take that with me? And the tongue movement as a control. If you look at several subjects we did so far about 6 subjects, but these are the 3 best. When you look at the foot, it's repetitively at around the same location. If you look at the penile shaft, it's somewhat more laterally located. And if you look at the foot on the right side, it's more medially located. We also ask the patient, or the volunteer in this case, to squeeze the pelvic floor. And you see that there is the supplementary somatic motor area activated and the primary motor area activated. Also at the same place, so repetitively possible. And if you look at the earlier experiments 20 years ago, and this is pelvic straining, and activation here is also more laterally, but it's different because here, during repetitive and ongoing straining, you see more medially activation. So it's different. The results are completely different than in individual subjects. With the whole brain method, you can also look at the cerebellum. Here's the foot activation, penile shaft, and the tongue area. So very nice somatotopic localization. I must say, it's rather tricky, the 7 Tesla MRI. If you want to do it yourself, work together with experienced neuroradiologists, but always be in the lead. Don't give the experiments to the neuroradiologist and get the data with you, because then you don't have any control over the design. Future method, maybe resting state functional MRI. Resting state functional MRI is the intrinsic activity of the brain in the absence of other stimuli. So you can do it maybe with a full bladder, scan the patient, and then put some kind of mark on it, whether he has severe overactive bladder or less overactive bladder, and you can link your treatment to it. It is also called spontaneous activity. An example was recently published. I find it a very interesting study. They did resting state functional MRI, Nardos and colleagues, and they said the connectivity between the areas may predict the severity of urinary urge incontinence. And they showed prefrontal areas are connected with areas more medially located in the cortex. But it's a very nice thought how we can go on in the future. The near future, we are building a prediction model for bladder floor dysfunction. And we use the technique of the Rotterdam Prostate Cancer Risk Calculator based on 200,000 patients, which is the main predictor model in prostate cancer. And the same people, Monique Robel and Ewout Steyenberg, who is in the world, in my view, one of the key figures in prediction models. They help me to make a model. 7 Tesla, I've shown, can detect very nicely changes in individual patients. Biomarker of bladder dysfunction can be detected in the individual patient and correlated with traditional parameters. Biomarkers have to be validated internally and externally in the prediction validation of its role in the clinical process, and that's very important. I can say, oh, my results are very nice, but if you don't validate that externally, it's always biased. All the results which I have spoken of the recent functional MRI in patients and earlier in volunteers, are biased towards the hypothesis. Take-home messages. Much progress has been made in the mechanism of action, but little clinical impact in my view. Better bladder dysfunction outcome needs a multimodal approach. Resting state functional MRI, maybe also diffusion tension imaging in single subjects with functional bladder dysfunction may have a clinical value. Brain biomarkers in single subjects have to be validated in a prediction outcome model on bladder and pelvic floor dysfunction. And very soon we will publish our first model on that. I want to acknowledge several people from Rotterdam: Sven Luiten, Johan Holstegen, Chris de Zeeuw from Neuroscience. From Urology, Sarah Reuvers, Lisette Hoen, Jeroen Schepen, Monique Robel and Jan Groen. From Amsterdam, the Spinoza High Field for Imaging, Wietske van der Zwaag. And internationally, people who cooperate with me in the last years, and I'm very grateful for them, for their loyalty. Jan Kroet, Limin Liao, Uli Meinert and Thomas Kessler. And to the Japanese people, I want to say domo arigato for the invitation, also to Professor Yoshimura. And I already know Professor Yoshimura for 25 years, and I will say our scientific journey, but also clinical journey, will go on. Thank you very much for your attention.
Thank you.